Hello, I'm Priya Lokre, Assistant Professor of Botany at Lugnete Ramdas Patil Duma Arts, Science and Commerce College, Rahuri. Let us see about the research topic. Study of diversity of coprophilous fungi from selected dung samples. The literally meaning of coprophilous fungi is the dung-loving fungi. Coprophilous represents the diverse community of morphological and physiologically specialized mycota which provides a biological force for the decomposition and recycling of animal feces. Many fungal conidia are ingested by the herbivorous animals while grazing, and due to that, coprophilous fungi exists in a broad range of habitats fulfilling significant role in the ecosystems. Dung contains a large quantity of readily available nutrients such as carbohydrates, high nitrogen content, vitamins, and growth factors. Aims and objective of our study: to survey the area selected for the collection of dung samples, to study the differences and similarities between fungal diversity of domestic and stray wastage, to study habitat diversity of coprophilous fungi occurring on both domestic and stray dung samples. And the last one is to isolate and identify the fungi from from dung samples. Here the main objective is the we have to observe or we have to uh, we have to observe the actual difference between the uh, domestic and stray animal wastage. Materials and methods. Selection of study area. As the dung is common herbivore waste, easily available, so the dung samples were collected from Srirampur, Rahuri and Sangamner Tehsil. The dung sample of cow, goat, horse were collected as the animals were easily available in the study area. Most of the samples were collected from different localities for the study of fungal diversity. Here, each dung sample was collected in a clean, airtight polythene bag and then taken into laboratory of Loknete Ramdas Patil Duma Arts, Science and Commerce College, Rauri, Department of Botany. Here, table number 1, common name of that animal, see its scientific name, uh, it is domestic or stray, and a location where samples was collected. So cow, horse and goat, three animals were selected. Isolation of fungi from the dung samples. The dung samples were subjected to isolation and enumeration of saprophytic and coprophilous fungi by moist chamber method. Each dung samples of domestic and stray animals was incubated in the moist chamber plates equidistantly with wide space for the growth of fungal mycelia. The samples were incubated for 5 to 10 days at 25 to 28 degrees Celsius, that is room temperature. The moist chamber plates don't need any special type of medium for the growth of saprophytic and coprophilous fungi on the dung samples. The fungi grows on their own host, that is dung. Identification of the isolated fungi. So preparation of fungal slides was made by putting the fungal mycelia and spores stained with cotton glue and then mounted in a lactophenol on slides. These slides then carefully observed under the uniocular compound microscope at different magnification, that is 45x 100x. So identification of isolated fungi was made on the morphological peculiarities such as color, texture and appearance. This is the result. There are altogether 12 types of fungi observed: Pilobolus, Parasola, Mucor, Rhizopus, Paniolus, Podospora, etc. And their presence and absence in domestic and stray type of animals in cow, horse and goat sequentially. These are some beautiful slides of fungi: Pilobolus, Podospora, Rhizopus. This is another Rhizopus slide. This is unidentified one. Okay, Chitomium. This is one unidentified one. Ascobolus, Chilimania, and this is the moist chamber. And look how beautifully the Pilobolus emerged out that from dung sample. This is the Pilobolus and mucor colony. Then Chitomium also. Here Ascobolus, Parasola, Panulus, and Coprinellus. So these are photographs which is taken from the actual spot also. Discussion. Pilobolus was found only domestic cow and goat dung samples, whereas Parasola was only found on domestic horse dung. Mucor is the only species which was found common in all dung samples selected for the study. Paniolus, Parasola, Coprinellus were found only on the domestic horse dung sample. The Podospora, Rhizopus, and Chilumenia were found only on the domestic cow dung sample. The Chitomium and Ascobolus were found only on the straight cow dung sample, and this study showed that the isolated species are distributed in three classes, that is Ascomycetes, Basidiomycetes, and Zygomycetes. Most of the isolated species belong to Ascomycetes, followed by Basidiomycetes and Zygomycetes. And this is the conclusion. So from this study it is concluded that animal dung is the good substrate for the production of coprophilous fungi. Total 12 species of coprophilous fungi were obtained, out of which 8 were microfungi and 4 were macrofungi. So considering the importance of these fungi, some are edible, some of them are poisonous, while some are enzyme producers. So they play an important role in the decomposition of organic matters, hence measuring soil fertility. Simply, coprophilous fungi are the nice organisms which are considered as important factor of the ecosystem. Lastly, this is a beautiful slide of. Thank you.